morning everyone. My name is Ridhima Majavdia from class 10 and today I, along with my friend Ridhima Thakur, am going to give you a deep insight on the topic of cloning.
So first of all, what is cloning?
Cloning is a technique scientists use to make exact genetic copies of living things. Genes, cells, tissues and even whole animals can be cloned. In biomedical research, cloning is broadly defined to mean the duplication of any kind of biological material for scientific study. Scientists often clone genes in order to study and better understand them.
Do you know how is cloning done?
So first of all, to clone a gene, researchers take DNA from a living organism and insert it into a carrier such as a bacteria or yeast. Every time that carrier reproduces, a new copy or a new clone of the gene is made.
How many types of animal cloning are there?
So there are two types of animal cloning. First is embryo twinning. Scientists first split an embryo in half. Those two halves are then placed in a mother's uterus. Those two halves, when they develop into a unique animal, the two animals share the same genes. The second process is somatic cell nuclear transfer, popularly called SCNT. It is a more popular method of cloning. So what happens is that to make a clone, scientists transfer the DNA from an animal somatic cell into an egg cell that has had its nucleus and DNA removed. The egg develops into an embryo that contains the same genes as the cell donor. Then the embryo is implanted into an adult female's uterus to grow.
And what is therapeutic cloning?
So therapeutic cloning is a very interesting process of cloning where the use of SCNT is done to reprogram somatic cells into undifferentiated cells for different therapeutic purposes such as the treatment of degenerative diseases or injuries or to correct genetically predisposed conditions. These cells, these undifferentiated cells, can also be used to treat a variety of conditions including Alzheimer's disease, Parkinson's disease, diabetes, mellitus, stroke and spinal cord injury. So, therapeutic cloning can be done in today's world to treat many diseases which cannot be cured in the past times.
Is there any experiments done in the field of cloning?
So there are many experiments that have been done in the field of cloning, but many of them turned out to be failures due to the complex mechanism involved in cloning. But one such experiment, the experiment done on Dolly the sheep, has turned out to be a very successful experiment. So Dolly is the female fin-dorsed sheep that lived from 1996 to 2003, and she is the first clone of an adult mammal produced by the British development biologist Ian Wilmot and colleagues of the Roslin Institute near Edinburgh, Scotland. The announcement in February 1997 in Dolly's birth marked a milestone of science, dispelling decades of presumption that mammals could not be cloned and igniting a debate concerning the many possible uses and misuses of mammalian cloning technology.
Can human cloning be done?
Absolutely, humans can also be cloned, but there are many ethical controversies involved in it. There are many psychological, social and physiological risks also involved. There are many failed attempts also, which makes the viable pregnancy not guaranteed. Because of the risk associated with the process of cloning, it introduces a very high likelihood of loss of life and the process is considered unethical.
Is there any laws for human cloning?
Yes, there is one such law. In 2005, the United Nations passed a non-binding declaration on human cloning that calls upon member states, and I quote, to adopt all measures necessary to prohibit all forms of cloning as they are incompatible with human dignity and the protection of human life. So even an organization such as United Nations recognizes the fact that human cloning, even though there is a future in human cloning, but it is considered unethical as it harms the dignity, equality and freedom of humans.
So what are the applications of animal cloning?
So even though human cloning is not considered a future, it is considered a future but is considered unethical, there's a very huge scope of animal cloning. First of all, the cloning of animals has been used in a number of different applications, such as to study diseases that develop in animals. It can also be used in agriculture where livestock of cows and pigs are cloned to give or to produce more milk or meat. Clones can even resurrect animals. For example, in 2001, a cat named Cece was the first pet to be created through cloning. Cloning might one day bring back extinct species like the woolly mammoth or giant panda. So, in conclusion, cloning can be used in many different ways in the field of animal cloning. Even though human cloning is considered unethical in today's world, it can be a life-changing approach in the field of genetic technology in the future. So, in today's world, we can take out many advantages of animal cloning, such as taking out cures of diseases, creating copies of animals for the field of agriculture, and much more. So, that's all on today's topic of cloning. This is me, Ridhima Majavdia and Ridhima Thakur signing off. Thank you.